Hello, how are you doing? Everyone, welcome, whether it's morning, afternoon, or evening. Welcome again to this exciting session on connectomics, which is the study of brain maps. And I'm so excited to be giving, kind of like a rundown of what neuroscience connectomics about. These brain maps allow us to understand different kind of organizational principles of the brain. If we understand how the structure of our brain is, then we can also understand its function and we can see if things goes wrong in our brain, which leads to some kind of disorders, right? So that's exciting. Okay, I'm gonna invite more people into the room. So as you can see here on the left, this is actually the brain of the Drosophila, the fly. And as you can see, we have a complete information in 2024 on over 140,000 neurons. And on the right is the human brain. And this is taken using the MRI. But because the human brain is so complex, as of this moment, we do not have a full information of the human brain yet. Full information, I mean like neuron to neuron, just because there are 60 billion neurons in the humans. And I'll be going over this, but I think it's just a cool image. Okay, I think the time is 11 a.m. in the UK, and I'm gonna get started. Thanks again to everyone for coming, and it's great to see you all here. So again, welcome to this workshop, Advanced Techniques in Exploring the Synapse-Level Connectome. And I know it says advanced technique, but it's actually meant for everyone who's interested in neuroscience to join this workshop. And some of you might have attended this workshop before. Hopefully, you get to learn something really cool. But if you're new to connectomics, this is also a chance for you to learn about this cutting edge field in neuroscience, which is very relevant to understanding the human mind and also in many of the neurological disorders and neuropsychiatry disorders as well. So hey everyone again, for those who just joined, I'm Sapo, a PhD student at the University of Oxford and co-chair of the 2025 FlyComp Research Competition. I use computational neuroscience to model the brain using the human connectome, but as you can see, the fly and the humans is they are different, but different animal models provide us different sources of information. And today I'll be leading workshop 4 on advanced techniques in exploring the synapse level connectome. Normally, I would be co-leading this workshop with another co-chair, Arushi Vardhan, who is also our research lead for FlyComp 2025. Unfortunately, she couldn't be here today for health reasons, but Arushi is actually, the real experts when it comes to fly connectomics, especially working with the tools that you will be using for the competition, which is Neuroglancer and neuPrint. So if you have attended our previous workshops, you would know that she eloquently demonstrates these tools and answers any questions regarding fly neuroscience. But the show must go on. And together, Arushi and I have put up some really exciting material for you all in today's session. And on her behalf, I will be demonstrating how to use Neuroglancer and neuPrint. Before we officially start, I would like to also thank Om, who helped us with outreach to high schools in Thailand. And also I thank the teachers and students from Sathit Chulalongkorn University Demonstration School and Kamnatwit, as well as our FlyComp 2025 ambassadors, partner clubs, and of course, thanking to you all for joining us today because the reason why our faculty partners, myself, Arushi, and everyone who is a part of this competition, our main mission is to inspire you to love neuroscience and also to show that from the comfort of your home, you can explore this amazing data set out there. So hopefully this session inspire you to explore the brain from the comfort of your home. So today, the session outline, I will be talking about I would give you some introduction to connectomics, and even if you have previously been into our workshop, it will be something new. And I will also explain a bit about the competition. I'll go very quickly because everything is up on the website. And then we'll be demonstrating tools like Neuroglancer and neuPrint. These tools are cutting edge connectomics tools to explore the brain, and you will be learning in this competition. Then we will go into a bit more advanced stuff, which is how do we actually build a computational brain model? And here I will be going a bit hardcore to physics, and some of the things might be really difficult to understand, but I'll try my best to break it down. I think that the point of this is to let you gain intuition. And I think gaining intuition is really important in physics and in neuroscience, because once you gain the intuition, the math part becomes just like a mirage, right? Like you in the end will get it because if you understand if you have an intuition for something, then the barriers to understanding the very difficult mathematical equation would kind of go away. And lastly, I'll be happy to answer any questions from the audience. So what is connectomics? So connectomics is the production and study of connectomes, and the connectome is really another fancy term for something called brain map. And you might be like, why do we need to spend so much time in creating these brain maps? And you will realize that if we don't have brain maps, we don't understand the brain. If we don't understand how our brain is organized, then we don't understand how we think, how we act, why we behave a certain way, or why do we get some kind of brain-based disorders like Parkinson's, dementia, depression, addiction, and so on. So that's why the study of brain map is kind of central to neuroscience. Here are some three reasons why connectomics matters a lot. First is because our mind emerges from how the brain organizes itself. So mental processes like perception, memory, emotion, decision making depend on how neurons are organized into circuits and networks. And you can also imagine that if something goes wrong in this organizational principle, then you get all sorts of neuropsychiatry disorders related to memory, emotion, decision making, and so on. So again, so here to reiterate, so disorders reflect disruptions of these organizational principles. And the disruptions could look in different ways depending on the type of analysis we are seeing. But an example is altered structural connectivity, which is, are there changes in how two different regions are connected to each other? Are there some kind of disrupted communications between brain regions? Or are there some kind of disintegration between large scale networks? The third important thing about connectomics is that connectomics bridge levels of explanation. Because we are trying to create brain maps, you can create brain maps at different levels. So you can have a brain map that incorporates transcriptomics, which is at the genetics level. The data set that you would be using is at a cellular, circuit, and network level. So essentially, you're having information about how two neurons connect with each other. So that's the cellular level. You also have information about neurotransmitter, which is kind of like the chemical messenger between two neurons. And because we have such a large scale comprehensive data set, you can infer some kind of circuits and network from this data set. And of course, the point of building these connectomes is that hopefully we can infer how these structural organization of the brain informs our behavior or our experience. So as you can see, connectomics bridge levels of explanation. So an example research question is how molecular change scale up to affect thoughts, emotions, and other cognitive processes. So what exactly are brain maps? And I know we have talked about this last time, but I'm going to give you a different perspective to this anyways. So what exactly are brain maps? So when you think of brain maps, you have to think of two things. The first is the scale. In other words, what's the level of detail we're mapping the brain? And there are two kind of scales. So one is the micro scale, where you're trying to map every single connections between individual neurons. So if you could imagine that in human brain we have 60 billion neurons and some trillions connections. I mean, right now it's almost impossible to do the entire thing for the humans. So that's why right now in the human we only have the macro scale, which is anything that's beyond the synaptic level. So if you take an MRI scan at the hospital, that's kind of like the macro scale. Anyways, the second point to consider when building a brain map is whether you're building a structural map or a functional map. A structural map is a map that conveys neuroanatomy of the brain, so it's the structure, right? And the functional map conveys the brain activity. In other words, the functional properties of the neurons. So, like how the neurons is firing, or in humans, is this brain area activated when we're trying to memorize something? So that's the functional brain map. So just keep that in mind, okay? You have scale, micro and macro, and then you have structure versus function. Okay, let's start with scale. So in scale, which is the level of detail, we have the synapse-level connectome. And this is actually the dataset that we will be doing for the competition. You would get a chance to explore a synapse-level connectome, and this beautiful looking image is of this fly brain. And if you know, the fly is so small, and it took us 20 years to be able to reconstruct this every single neural connection of this very tiny brain. And this is so small, literally. It's smaller than, I think it's less than 1 millimeter cube. So it's just like really, really tiny. And it took us 20 years to get here. So it's amazing. It's released last year. And now you as a high school graduate students get to explore this data set together. So Fly Connectomics, why are we using Fly is because right now in the synapse level. This is one of the most comprehensive synapse level connectome we have. And as you can see, I've put a bunch of beautiful images of different parts of the brain. But specifically for our competition, we'll be looking at this part. So this thing here on the right. So this is called the optical lobe. And as you can see, it occupies over half of the fly brain. And this region is called the optic lobe because it's got to do with the fly visual system. And you're like, why do we have to care about vision? But actually, fly, due to evolution, the eye of the fly is kind of one of the most important organ in the fly. And because a lot of things rely on the vision, right? Like imagine the fly comes on your food and you try to shoo it away, it actually escapes really quickly. Why? Is because half of its brain literally goes into motion detection and detecting a bunch of visual features, which in turn leads to some kind of behavioral response. So you will see flight connectomics is quite cool, and in some sense allows us to understand a lot of cognitive process that goes on in human as well. Okay, here are some other synapse-level connectomes in other species. Recently in April, there's this Microns project. This is a beautiful image from the mouse visual cortex. So as you can see here, it is an unprecedented dataset of high-resolution anatomical images of individual cells in mouse visual cortex mapped onto their response. So we had the fly, so the fly is of the entire brain, and for the competition, we're using an optic lobe dataset, which is again almost half of its brain, pretty much. This is of the mice, but because the mice brain is so huge, this is like very tiny part of the mice. Again, we are still far from actually have a complete synapse level connectome of the mice, but we will get there someday, maybe 5 years down the line. So this is really cutting edge stuff. Then we have the humans. And you could imagine that in humans, right now, the best we have is 1 millimeter cube of this tiny part of the brain called the temporal cortex, and just 1 millimeter cube alone. So that's kind of like a few grain of sand, pretty much. You have 1.4 petabytes of microscopy data. This 1 millimeter cube of this brain has over 150 million synapses, which is the connections, right? And 57,000 cells. And can you imagine just 1 millimeter cube of the human brain has so much of data already? So to scale it up to the entire brain would probably take some time. I'm sure we will hopefully have a full synapse level human brain connectome at some point, but that will be 10 years down the line. But at this moment, the fly brain is one of the most comprehensive wiring diagram, and that's why we chose to use the fly connectome for you guys to explore. But anyways, just to give you an intuition of how do we even reconstruct these kind of beautiful images, so I want to go with you step by step. But before that, I want to give you a teaser of what we are going to be using for the competition. So when you're exploring the synapse level connectome, there are two kind of tools that we normally use. The first tool is called Neuroglancer. So Neuroglancer is a web-based browser tool that allows you to visualize the neuron. And you can visualize individual neurons, and I'm going to demonstrate it for you. But as you can see, you have different kind of functions that you can do with Neuroglancer. So for example, C2 is a neuron type. So neuron type, you could think of like if you go to a supermarket, you see green lace. Green lace is a neuron type. And if you go to, let's say, Tesco, you can see 20 green lace chips on a shelf. So as you can see, you have many C2 neurons as well in the brain. Then you have neurotransmitter activity for that particular neuron type. So neurotransmitter is the chemical that's sent between two neurons, and the neurotransmitter can either inhibit the next neuron. So inhibit means that it reduces the chances that the next neuron will fire, or the neurotransmitter can be excitatory. So excitatory neurotransmitter are the neurotransmitters that increase the probability that the neuron that it projects to is going to fire. You can also look at the synaptic sites. So you can look at where this neuron is receiving inputs and sending out inputs. So post-syn stands for post-synaptic sites. So remember, all of this is in the perspective of this particular neuron. So in blue, this particular neuron, so in blue, this is a post-synaptic site for the neuron C2. In other words, in these blue regions, it's receiving inputs, it's receiving incoming information. And in red, this is where it's sending out information to the other neurons. The other tools that I'll be demonstrating today is called neuPrint. So neuPrint is a very complex database kind of thing, but it's very powerful because we can query neuronal connectivity. So for instance, for the C2 neuron, you can see how many synapses it's receiving as its inputs and how many presynaptic sites or how many sites are there that sends information away. It also gives you some kind of brain region breakdown as to like which brain region does this neuron receives information from. So ME. So again, we'll go over this, but just to give you a kind of teaser. Okay, there are two main things to consider. So we considered the synapse level connectome. Now let's talk about macro scale. So the macro scale connectome, it's done in many species, but of particular importance of humans. Why? Because, like I said before, humans have 60 billion neurons, so we don't have a microscale map yet for humans. So that's why right now we only have the macroscale map of the whole brain for humans. But one of the project is called the Human Connectome Project. So the ideal Human Connectome Project is to tackle one of the greatest scientific challenges of the 21st century, which is mapping the human brain, and it aims to connect its structure to function and behavior. But Human Connectome Project, essentially, you get like thousands of people to do MRI scans, and together you can combine them to get a beautiful map like this. But again, both microscale and macroscale have a unifying goal, which is to connect a structure to function and behavior. And regardless of the scale, the principle of connectomics remain very similar. So here I want to kind of compare and contrast between the macro scale and the micro scale, right? So this micro scale is essentially this one that I showed you. So I'm going to explain how micro scale connectome is normally being reconstructed. I've kind of broken this down into more simpler terms from the actual paper in Science. So, and again, some of these things might be a bit difficult, but just gain an intuition. And if you have any questions, like please type in the chat. Don't be afraid. This is the whole brain. But how do we get a microscale connectome from a piece of that brain? So essentially what you do is, like our brain is how many centimeters? I don't know, but it's definitely more than 4.5 millimeters square. And so you essentially take a very small part of this brain, 4.5 millimeters square. It's not even centimeter, right? It's millimeter square. And you get kind of like this. So when you take a cube of 4.5 millimeters square of live human brain tissue, you kind of dye this tissue. So dye means you try to color the tissue so that different cell types within this particular slice of the brain tissue is colored. And that's how you get like these beautiful images you see online. Then they slice this brain into extremely thin sheets, 33 nanometers thick, just imagine what is 33 nanometers thick. Our human hair is 80,000 nanometers thick. And now scientists, because our neurons are so dense, they have to slice it to 33 nanometers. This is so, so, so small. It's way thinner than a human hair. And then each of these slices is then picked up onto a long tape. So use some kind of very special tape to pick up this 33 nanometer thick slice. Then they photographed each slice with a special kind of electron microscope. Then they took a lot of overlapping pictures or tiles and they stitched them together. So here, as you can see, they take from this. So this is 3 millimeter or 750,000 pixel. This is 2 millimeter or 500,000 pixel of layer 1 of the human brain. Then you take this very small section of this 4.5 or 6 millimeters square kind of brain. Then you take very small images like overlapping tiles. And then for this particular image, you kind of get like some kind of image like this. Okay, see? Very cool stuff. So each tiny dot or pixel in the picture represents a 4 nanometer by 4 nanometer square, small enough to resolve synapses. Remember, the brain is so dense, we need to go that micro scale. And then you repeat this process many, many times, and then you stack the images together. And then when you stack the images together, you then trace the neuron using some kind of very fancy machine learning algorithm, which is out of scope for today's presentation, but you can go in our previous workshops, which we kind of covered the flood filling networks and some sort of algorithm to reconstruct these neurons. But hopefully you appreciate how difficult it is to reconstruct this human brain at a micro scale, or even the fly brain. It uses the same technique. And with that, we now have, as you can see, this is the cube of the brain, and you can sort of like reconstruct beautiful images of all sorts of neurons. We have a chat. Amazing. Excellent. Feel free to engage in the chat. I'll be looking at it. I would love to have you guys engage because I think it makes the conversation a bit more fun. So let's do a short quiz. So now time for you to engage. So here, remember what the scale of structural versus functional? I want you guys to guess if this image you see here is to do with structure or to do with function. If you could type in the chat, that would be excellent. Wipu says it's function. Anybody else? Come on. Would you like to say why it's function? Why is this functional? Why is this not structure? See, we have a brain map here too. Great. So of course the answer is function, but in case you're confused as to why this function, because you see, you take the MRI scan, this is function because we're trying to see Yes, someone wrote a message to me. It measures the brain activity. That's correct. So this is actually a type of MRI called functional MRI, and it uses the oxygenated blood flow and it infers activity from the oxygenated blood flow into the brain. And these blue and red are simply changes in the activity or in the signal over time. And you can have like different areas and you can, so let's say this yellow thing probably belongs to a brain region. This blue thing probably belongs to another brain region, and you can have like some kind of oscillating brain activities over time. But great job. This is functional. And do you think this is a micro scale or macro scale? Okay, someone texted me saying is macro. Great, it seems like most of you got it right. This is a macro scale. Why? Because remember, we are not resolving at the level of synapse. And this is how most human neuroimaging studies are done. You look at the macro scale. Yes, that's right. Macro scale because it's above synaptic level. Excellent. Great job, guys. I'm very happy. And so this is a macro scale. Do you think this is structure or functional? Feel free to type in the chat, but I want to explain this beautiful image. So this is a brain recording of a zebrafish. So someone said this is structural. Okay, so keep typing in the chat. What do you think? Is this structural or is this functional? Okay, and someone said functional. So functional, great. So just to give you context, this one is recording. Wait, I want to have this image to keep going. Excellent. I love this orange thing. But essentially, you're recording the brain of zebrafish at rest. So in other words, when it's not doing anything. And these orange blobs belongs to activation, like some kind of brain activation in the zebrafish. And you go, whoa, it's suddenly the entire brain synchronizes at the same time and activates. So because this is a recording of the brain activity of zebrafish, this is actually another functional map. Now is this micro scale or macro scale? Anyone? I've received answers. I see some folks attended a previous workshop. We have used this. Yes, the answer is micro scale. Good job, everyone. All of you got it right. And this is micro because we are using a microscopy. So actually, these brain activations at the level of individual neurons. So if you go look at this original paper in Nature Methods, you can see they were able to resolve the brain activity at the neuronal level. Excellent. So I'll move on. So this is micro scale. Great job. And I want to show you this because remember the second point we need to consider with structure versus function. So these two examples I've shown you, they are called functional imaging. And here it's functional because we are recording neuronal activities. And it could be at a more macro scale like in humans, or it could be more at a micro scale like for zebrafish. And we actually have the functional imaging for the fly as well, and we have it for different types of species. Excellent. Now, what is the competition about? Like I said before, scientists very recently have created one of the most comprehensive brain maps that exists out there, and today you will be exploring not just today actually, for until December 1st, you will be able to explore one of the most detailed brain maps ever created. And it's hosted by my nonprofit called Clematis, which aims to empower next generation of researchers. And hopefully we have made this as accessible as it can be. And we are excited to partner with our faculty partners. They are Gruntman Labs and Ryder Labs at the University of Toronto. We also partner with pedagogical experts. His name is Professor Steve Jordans, and he specializes in educational psychology. So as you can see all of our resources, we try our best to make sure that even you as a high school student or undergraduate student can explore this dataset and write a nice report, which I'll go over it. And also thank you to our student partners who actually help collaborated with us to make sure that this initiative is accessible to everyone. And of course, thanks to you all for being here. And as always, you're welcome to give feedback on anything that we are doing. So what is this competition about? So you will be guided by neuroscientists, including me, including Arushi, and we have awesome guest speakers as well. But the idea is that you will be using the two tools, the Neuroglancer and the neuPrint. And by the way, the image on the right is actually of the fly brain. You see, it's just beautiful stuff. We are going to use Neuroglancer. So like I said, Neuroglancer is a 3D visualization tools of volumetric brain data. Volumetric because the brain is in volumes. And we're going to use neuPrint to query neuronal connectivity. And what's the challenge? The challenge is you're going to choose a neuron in this brain. There's tons of neurons to choose from, and you can choose it from Neuroglancer, which I'm going to show you, or you can choose from the literature as well, as long as it belongs in this optic lobe dataset. Then you're going to explore neuron morphology. So you're going to explore what is the structure of this neuron, and I'm going to demonstrate today. Then you're going to analyze neuronal connectivity using neuPrint. And then lastly, you're going to propose how your neuron process information and contributes to fly behavior. So remember, the goal of this is that you use connectomic analysis. So you visualize the structure, you look at the connectivity, then you go look at the literature, and hopefully you can come up with something interesting of how your connectomics analysis have allowed you to propose something of how a certain neuron process information within a context of network kind of thing, and how it contributes to a certain behavior. And this will result in two final deliverables. The first is a creative piece. So the creative piece is meant to for you to practice scientific communication because as a scientist, or even if you, let's say, decide to do medicine or any other careers, really, you have to learn how to talk about your work. And your work will be about you will basically try to communicate to the public or why is this neuron so cool? Why did connectomics analysis so crucial to your work? And if you can inspire other students to join our mission to also explore more brain stuff, then that's excellent. So that's creative piece. And you can do poster, art, infographic, videos. We have amazing submissions already. So the point is you can be creative as you want. Then you also have a written research report. This is 500 to 1,000 words of research report. And again, all of this can be viewed in our website. The submission deadline is December 1st, and we announce the winners by January 2026. We also have 500 Canadian dollars in prizes, and you can look all of that up in the website. And high school students have separate prize categories from the undergraduate students, so try your best. Anyways, but the point of this competition is really for you to gain real research experience in using the neurons. So I'm going to demo a neuron example called T4A, but just to show you how connectomic analysis journey might look like in Neuroglancer and neuPrint. So the first step in Neuroglancer is to see something called ROI. So ROI stands for region of interest. So you need to see the region interest to see which neuropils your neurons has its arbors in. A bunch of terms, but essentially the fly brain or the dataset that we are concerned with have four main regions. In the fly literature, we call these neuropils. Okay, so we have the first one called lamina. When the fly sees the world, it receives light input through the eye, like photoreceptors, and then it projects the first thing to lamina. Then it goes to medulla. This medulla is kind of like a big kind of region, and it houses a lot of cool neurons. And then it sends the projections to either the lobula, which is another brain area here in green, and the nodular plate, which is the another brain area. And when you actually go in the Neuroglancer, you won't have these labels, but you have these colors. So oftentimes it's nice to kind of look back and try to memorize the brain region so that you can kind of describe the neuroanatomy of the neurons really well. Let's try and go to the Neuroglancer. So this is the website, by the way. Actually, I can just send this right now on the chat. This website has a bunch of cool things. So it explains, please join our mailing list because it's where we send out very cool resources. And we have this handbook. So we have the guidebook, we call it the handbook. So the handbook has every single thing that you need to submit the report. So including like what your research project would be about, how to submit the things, what are the deadlines, selection criteria, and so on. So I suggest you read this. Then we also have starter guide for those who are very new to neuroscience. We explain what a dendrite is and so on, neurotransmitters, the optic lobe. And again, a lot of these are actually covered in the previous workshop as well. But importantly, we have user guide for Neuroglancer. So we have like a bunch of screenshots and so on. So please have a look. We also have a research report template. We kind of try to create a guide as to how you can write a nice report. And lastly, we have a learning guide. So a lot of students have actually asked us when we launched the competition, like, this sounds very difficult. What do I need to know? The neuroscience so vast. So here we kind of provide like you can think of like a syllabus, and we try to map it to, let's say, like a curriculum that you might be studying. So a lot of undergraduate and high school curriculums in biology would require you to know these anyways, like what a neuron. So you kind of need to know this. And we have like some suggested resources where you can go. So essentially, the point of learning guide is that if you like don't know anything, or even if you know a lot of things, it really doesn't matter. We try to create an equal playing field. As long as you know these concepts in neuroscience, then you should all be equally able to write the report. So that's the point of the learning guide. But anyways, so you can click here. So use powerful connectomics tools and you can click this link, a Neuroglancer. So the link sends you to an optic lobe dataset. It takes some time to load, but hey, that's a fly brain. There we go. We have neuropil shell. So these are shell. Then we want to open something called RI. So we want to open this up. Yeah. So now you might be very familiar, but remember the eyes are kind of like this, right? This is the lamina, so you can rotate. You should play around. It's all in the guide, but you can rotate up and down. So let's go up. So maybe this might be a bit easier for you to see. So we can play around, but essentially we have the lamina, the medulla, the lobula, and the lobular plate. And as you can see here, lamina, medulla, lobular plate. The way Arushi normally explains is quite funny, but so because lamina is incomplete, it kind of looks like that. The medulla is in the middle, so middle medulla, though it's got nothing to do with the word middle, but it's kind of the largest thing here. And we have the lobula and the lobular plate. Apparently, the lobular plate looks like a plate because it's a bit thinner, the way I remember it. Because remember, I'm new to FlyComp as much as you. So the lobular plate is this purple thing and it kind of looks like a plate. So now we know what these regions of interest are. So now let's try to pick some neurons. So we did say we want a T4A neuron, but essentially these are neuron types that you can see. So we have, oh, C2 neuron. You have C3 and so on. You can choose any neuron you want. But let's try with T4A. So T4A, let's pick a neuron that would show a nice color. Would red work? Let's try red. Okay, so as you can see, we have tons of T4A neurons, but each of these neurons, so there are many T4As, and these are differentiated by the IDE, but you can choose anyone. So the first step is to pick a neuron. So let's say I picked the T4A neuron. Great. So now you try to rotate around and you try to look at the structure of this neuron. So as you can see in this neuron, you have kind of like the cell body, the dendrites, and so on. But an important thing is you try to click presyn and postsyn. So these are the synaptic sites. So as a reminder, post synapses receives incoming information and presyn receives outgoing information. So that's what it is. And then the third is to be able to describe the layers. So you want to see which, so layers is a way to describe where is it in space. So let's try and look in terms of layers. So for this T4A, you can see that the neurons kind of cut through two different regions. So it cuts through the medulla. It also cuts through the lobular plate, the purple part, as you can see here. I'm trying to go slowly. Yeah, here we go. So let's try and look at in which layer is this cutting at the lobular plate. So the way you would go is you would click LO. So these are the acronyms: lobular plate, medulla. And you can look at these acronyms in our slides or in our guide. It has all this information. Also, I'm gonna try and go over this a bit quickly because we have presented in the previous workshop. So if you are really confused, please look at the guide or our previous workshops on our YouTube. But to quickly show, so because we know that this particular neuron is in this purple region, we're gonna click lobular plate layers. So we click that, and the next step is to actually click on this show layer side panel. That's what it is. Here we go. So the lobular plate have 4 layers, and I'm gonna just open it at once. And it's gonna take some time to load. But as you can see, these are kind of divided like that in different colors. So now, as you can see, it covered all of our synaptic sites in the lobular plate. So again, we're looking at the lobular plate. Now the question is we want to know which layers do these synaptic sites lie in the lobular plate. So the way we're gonna do is we're gonna unpeel one by one. So let's peel off layer 1. As you can see, when you peel off layer 1, you see immediately that it's actually layer 1 covering this thing. So let's put it back again. See? If you put layer 1, you actually cannot see any of the neurons. But now you take off layer 1, you're like, oh wow, I can see it now. I can see these sites. So what this means is that this particular neuron is passing through layer 1 of the lobular plate for sure. So now I'm gonna peel from the other side, so from layer 4. Look, so from layer 4, when you peel them off, so you click this I button and you peel them off, you actually cannot see any of the synaptic sites. So what that means is that this neuron doesn't really integrate information or send information in layer 4, so it doesn't matter. So let's 3-layer see. See, in layer 3, we also do not have any of the presynaptic or postsynaptic sites. Now let's look at layer 2. Look again at layer 2, you cannot see any of these sites. So what this means is that, which neuron is this? T4A neuron. This T4A neuron, it passes through the layer 1 of the lobular plate. So this is a way that we describe the morphology, and you should absolutely write it in your report. And hopefully that demos how to see the layers. So here we have the layers. This is another neuron, which is in the medulla, I guess. So now we're going to a neuPrint to query neuronal connectivity. So we're going to click a neuPrint, take it some time to load, but essentially you can look at a neuron of interest. So we were seeing T4A. So let's look at T4A, and you can just click submit. Okay, so you type and then you click submit. Here, and by the way, you need a Google account, so just type your Gmail or something. So you see you have a bunch of T4A, but again, it doesn't really matter. So you're going to look at a random one. As you can see here, T4A neuron ID 100018 have 261 postsynaptic sites, which is where it receives input, and it has 93 sites that sends output, or presynaptic sites. Then there's a brain region breakdown. It receives input mostly in the medulla, so ME stands for medulla, and it sends output mostly to the areas in the lobular plate, so LOP. And then you can also see the consensus neurotransmitter. So this is acetylcholine. So we know that this particular neurons, it sends acetylcholine. So it sends acetylcholine to other regions. Anyways, but the point is we can pick any random neuron that you want of T4A. And then we're going to click this C. The C button is basically like a circular chart. It's synaptic connectivity. So let's click that. Here we go. So for this particular neuron, it receives upstream. So if I click on upstream, so you can actually tell from where does it receive the upstream. So this particular neuron receives upstream from MI1, from TM3, and all of these are at the medulla. It also receives input from Y13, Y11, and so on. And the larger this section takes, it means that MI1 is the major input to this particular neuron. And let's look at downstream. So you can see downstream, it also kind of sends its output to a neuron in the lobular plate, more actually. So that's interesting, right? So it sends a lot of output to LPI12, LPC1, and so on. And there are a bunch of neurons in the fly, but that's the point of the neuPrint. You can now explore neuronal connectivity in the brain. And I'm going to show you, this is now outside the scope of the competition, but in case you're curious, you can actually change query type, and then you can go to rank table. And let's type again T4A. So you can actually create a rank for T4A to which of these neurons is sending the output. So you can click submit. Sometimes neuPrint, it takes some time, but that's okay. Let's see if I have something preloaded up. So I'm going to do that again. So we go to search, we change query type, you go to general, you click rank table, type T4A, you click submit. Oh, here we go. It just takes some time. I think it's because T4A has so many connections. If we look at this particular neuron of T4A, you can see that it sends. Okay, since it's working, I'm just gonna wait, but it seems like my neuPrint is crashing, which is fine. But you can see that this particular T4A neuron sends its output to LPI12R the most, and so on. So this is just another way of seeing the thing. No, come on. Okay, great. So now we're gonna come back to our presentation. So hopefully so far you have understood how to use Neuroglancer and neuPrint. And again, keep in mind that you're more than welcome to explore our previous workshop, which we have covered very much in detail. So I'm gonna put this laser pointer on back again. So now I want to go into a bit more computational neuroscience, which is actually what I plan to do. The focus of this talk is to show how useful is a connectome? So how useful is this information, or specifically the data set that we're going to be using? So I want to allow you to gain an intuition on how do you build an in silico brain model. Now, you don't need to know right now what in silico is, but just know that we are now trying to build some kind of computational brain model. And I want you to gain an intuition of how this works. So in other words, can we simulate the brain activity on a computer? So when you do something in a computer, This is called in silico. So in other words, can we create like a kind of like functional brain map that's defined by some kind of mathematical description? So that is what in silico means. And the point of having in silico model is so that we can now mathematically change the brain. So instead of doing experiments, we can just change things mathematically in our virtual model, and you can do some kind of hypothesis and prediction and so on. Of course, for the competition, you will not need to develop computational brain model for the competition, but I want you to appreciate that if down the line you're interested in neuroscience or even the application of neuroscience in medicine, this is very cool cutting edge stuff. So a lot of stuff might go over your head, but just gain an intuition, okay? This is the point of the talk. An example is our next guest speaker's work. So his name is Dr. Srinit Turaga. He's a principal investigator or group leader at HHMI Genelia. So HHMI is a Howard Hughes Medical Institute. You might not know, but a lot of times these very high risk projects, such as funding this connectome, needs a lot of money. And so they have institutions like HHMI, which is one of the largest endowed medical institution or charity that funds cutting edge biomedical research or bioscience research in the United States. And they basically funded this data set. And HHMI Genelia is a unique research campus where the principal investigators do cutting edge research with a very tight knit group, and it's an amazing place to go if you're interested in neuroscience. So, Dr. Shinitora recently uses the dataset that we actually will be exploring to answer the question, can we simulate a fly connectome to predict neuroactivity? So the title of this paper is called Connectome-Constrained Networks Predict Neuroactivity Across the Fly Visual System. It's published in Nature, and he will be giving us a talk on September 25th, 10 a.m. Toronto time, which will be 3 p.m. in the UK and 9 p.m. in Indo-China time. But I want to give you a glimpse of what it's like to build a in silico model, and then we'll come back to this. There are some key steps to build an in silico brain model. But the first information we really need is that we need to have a real anatomical brain map. And this can be any kind of map, but because for the fly, we have such detailed information, so we're going to use the synapse-level fly connectome, just because it has so much information, a very detailed wiring diagram of the brain. Then the next step is we are going to use principles of physics to simulate how a neuron would fire. In other words, we're going to use principle of physics to simulate the brain activity from this dead brain map. Remember, this real anatomical brain map is from a dead fly brain. But now, because we know how neurons fire, like we understand how neuron actually fires in response to like inputs from other neurons. So we're going to use principle of physics to kind of mathematically describe this. So when we are doing something like this is called building a computational model of brain activity, or in other words, we are building an in silico brain model. So how do we get a real anatomical brain map? So of course we can use the fly brain. So this left side here is the fly brain. For you, this is the composition. So we're going to use the fly connectome for the composition. But for the right side here is the human brain. But essentially the framework remains the same. You can take any brain map depending on the type of analysis you want to do. So why do we need a real anatomical brain map? It's because we need information on how two neurons connect. So for a micro scale, you can tell that neuron A perhaps sends information to neuron B through this intermediary neuron. And then you can also see that this white circle sends an inhibitory output to another neuron. And you can see that some neurons are only unidirectional, excitatory unidirectional. Some of them are reciprocal, like kind of like this neuron A and B is forming a reciprocal loop. So these information are really helpful when we're trying to create a mathematical model of how the neurons would fire in response to in this connectomic framework. And you can get these connectivity information from the neuPrint, which is the tools that you will be using. And as you can see, neuPrint, you can see sort of like the upstream and the downstream of the neurons. You can also visualize the synaptic sites. You can also see how many synaptic sites are there, like how many inputs receiving and how much output is sending. In the human brain, it's again very similar, but because we do not have information between two neurons, instead you're looking at a more of like a regional kind of analysis. So essentially, if you look at this brain area in the right, so each of these areas represent a brain area, so like a node. And then you can kind of create a nice structural connectivity map as to how strong are the connections between these two regions. So to get this visualization, you kind of need to have a graph. So in this graph, I want to allow you to have an intuition, but essentially each row here and each column here represents a brain region. So brain region A, brain region B, and so on to brain region T. So let's try to look at this particular one, right? Like this one. So now we want to see how strong are the structural connections between region B and region C. So as you can see, it's quite dark. So that means there's a very strong connection. So anyways, so now we get so from this map, you can then infer some kind of like structural connectivity between these regions. So an example is this region, this red dot here might be B. This region might be C. And this particular red line would be represented as a dot here in this graph. But anyways, the idea is the same. A real analytical brain map gives information as to how two neurons or two regions connect with each other. That's about it. And then as a reminder, we're gonna now take the information from the real anatomical brain map to simulate how the neuron would fire, or to simulate how populations of neuron would fire in terms of like the macro scale data set. And this is basically a way to build a computer model of the brain activity. An example is this scary looking equation, but actually, although you might not understand every bit of this, I think what you can take away is an intuition of how these equations work. And so let's go one by one. So I know this looks very complex, but this is essentially a simplified mathematical model of how neurons integrate inputs over time. So this equation is what Dr. Toraga used to model the neuronal dynamics. So I'm going to try to explain this so that you can gain an intuition. When I said simplified, it does not mean that I have simplified this equation. So this equation, it is as it is. But when it says simplified, remember the neuron is so complex. You have thousands of parameters that could influence how a neuron would fire. But instead, what a lot of mathematicians do is we try to find the most, not always, but sometimes a simple computational mathematical description that kind of accurately describes how the neuron would fire, even though it doesn't take account of everything, but largely it can model how the real neuron would work. So this is what simplified means. And in modeling, we always try to simplify things. So there's a balance between making a simple model, simple equations, versus how well does it really mimic the real brain or the real neuron. But anyway, so that's what simplified means. So in this equation, first thing is we need to care about VI. So VI stands for the voltage of neuron. So remember in a neuron, it's to do with the membrane potential or the voltage of the neuron. So if you can think of like a battery, right? A battery with higher voltage would sort of make the light bulb it would be brighter. So same thing with the voltage. Of course, there's very complex things, but here it makes a very reasonable and simple assumption in that if there's a higher voltage in the neuron, it means that the current neuronal activity would be higher. In other words, at this moment in time, the VI determines the neuronal activity. So with higher V means there's higher activity in this neuron, and I is the neuron. So in other words, this is the activity of neuron I at this point in time. Hopefully that makes sense. Okay, an important part of neurophysiology is to see the time constant and to see the rate of change of the neuron activity. So this is tau. So this is a time constant. An intuition to get at what time constant is the reaction speed of this neuron. In other words, if you have small value of this, it means that the activity level of neuron changes almost instantly. It changes almost instantly to any input it gets from other neurons. It's very sensitive to how other neurons communicating with this. But if you have big time constant, the activity level changes relatively slowly. In other words, the activity neuron is not that much sensitive to kind of like other things. This part of the equation explains the rate of change of neuronal activity. So it's just a way of saying how quickly the neuron I activity level is changing per unit time. Okay, so that's all you need to know. So this part of the equation explains, it essentially means the total synaptic input from neuron J. So remember now we're trying to find the connectivity between I and J. So we have the neuron I, but I want to see like how much information is the neuron J sending to this neuron I. And if you think about it, neuron J can be excitatory or it can be inhibitory. So this is where neurotransmitter is important. So if you have, let's say, a GABA neurotransmitter that neuron J sends to neuron I, the neuron J would essentially reduces the current activity, which is the VI. So as you can see, that each connection between I and J could either push the current activity up or down, depending on the type of neurotransmitter. So now let's look at this part of the equation. So this part of the equation, so remember VI stands for the current activity of the neuron. Current stands for the activity of neuron at this point in time. Now VREST stands for activity at rest. So as you might know that our brain, even when we sleep or even when we do not do anything, it still fires anyways. So every neuron has an intrinsic property in that it kind of has a resting membrane potential. But just to give you an intuition, just think of it like an activity at rest, right? It's kind of like a baseline. Technically it's not, but for now, let's just assume that it's activity at baseline. And what's the point of this? So because this model is trying to create a mathematical description of how a neuron fires, essentially, if you think of a neuron, it receives an input from, I don't know, from neuron J. So when it receives an excitatory input, the voltage spikes up. But the thing is, as the voltage keeps moving upwards or keeps moving above the baseline, this mathematical model essentially tries to show that there's always a tendency to keep going back to the baseline. So essentially, as soon as a neuron has this higher activity, because of the nature of the neuron, it always pulls back to the baseline. So it's described by this minus bracket, current activity minus baseline here. So anyways, but it really doesn't matter for to gain intuition. I just want to know like these equations can be understandable. But why did I show you those equations? Is because these mathematical equations that model neuronal firing have parameters. Those variables that we were seeing, these are called parameters. So each of these are parameters. Now, some parameters are known, so we know information on some of these parameters, but some parameters are unknown. And for those of the unknown parameters, we assign them with some initial values. Okay? So an example of what is a known parameter. Remember, a lot of time when building a brain model, we need information from the structural connectome or from the brain map. So from the structure connectome, we have information about how neurons connect to each other. So an example is that, like let's say from the FlyComp 2025 dataset, we have information on the number of synapses that neuron I receives from neuron J, which is essentially this part of the equation. So you see in this modeling of ours, we have an almost complete information of this, which is a great thing. So that's a known parameter. But what's unknown? So let's see the unknown. So just to repeat, so we know this total synaptic input from neuron J, and therefore, for this particular parameter, which is total synaptic input from neuron J, we would directly plug in the value from our structural connectome. So let's say we're looking at T4A as the neuron, and as we know, it receives input from, let's say, some kind of neuron from the lobular plate, right? So LPL something. So that would be the neuron J. So you can essentially calculate the total synaptic input that the neuron J from the lobular plate would send to the T4, just using the neuPrint. But remember, our goal is to build a computational model that accurately describes the real brain. But before we start refining the model, we first initialize the unknown or free parameters with some reasonable initial values. In other words, what I'm trying to say is that the point of building this model is we can hopefully have a nice description of the brain. But because a lot of things are unknown, we need to fine tune this model, right? But to fine tune this model properly, we need to start from somewhere. So this is the process of initialization. So you would try to initialize this unknown or free parameters with some reasonable initial values. But first, let's try to talk about what are some of the unknown parameters if we were to use the Fly Connectome dataset for our composition. Okay? The first thing here is the intrinsic properties of the neuron. So the activity at rest, it depends on the membrane property of that particular neuron, right? And this is something that the structure connectome cannot tell us. The structure connectome, all it does is it tells us the connectivity, but it doesn't tell you the intrinsic functional property of this particular neuron I. So that's why here, this is the unknown parameter. Another thing here, Remember what the time constant is? The time constant is the reaction speed. In other words, how quickly is this neuron responding to some kind of change. So if we look here, if we have small time constant, the activity level kind of changes almost instantly. And this is determined again by the membrane property. If you model membrane as a capacitor, if you're interested in physics, you can go look up like how the neuronal membrane is essentially a capacitor. And the tau or the time constant is intrinsic to the membrane property. Again, we do not know about this from the structured connectome. And if that's all we have, these two are essentially the unknown parameters. I hope that makes sense. Okay, so remember I said we need to first initialize some kind of values. So here from Dr. Turaga's neural network model that he's creating, he initializes initial time constant at 50 milliseconds. And then for this resting membrane potential, resting voltage potential. It's the same thing. And to initialize the resting membrane potential, he uses some kind of Gaussian distribution. I know this sounds a bit sophisticated, but it really not. Essentially, there are cool statisticians out there who essentially have mathematical description of different distribution. Because in reality, in our brain, every neuron have somewhat different membrane. They have different resting membrane potential. So we don't want to keep everything the same. The way it does is that it kind of creates a random resting state membrane potential using some kind of statistical distribution where the mean of the activity is 0.5 and the variance or like the standard deviation is 0.05. So you essentially create a random activity at rest for these neurons, essentially, where the mean activity of this random, if you combine all of the populations of neurons of the activity together, you would see that the mean is 0.5 and the variance is 0.05. And they're doing it in random units. Essentially, we are not really using the real membrane potential, which is around like minus 70 millivolts or something like that. But instead, they use a random units where 0 means no activity and 1 means maximal activity. So you're creating that kind of distribution there. But it really doesn't matter for the purpose of this talk. Just know that the point of this is for the unknown free parameters. We are, initializing some kind of values. Now, once we initialize these free parameters, we then try to refine our computer brain model by changing these parameters until our simulated model, so the model that's from the mat, roughly equals the real experimental data. Once we do that, we now have a mathematical description of how our brain works. If you look at humans, look at flies, look at mouse, look at zebrafish, you look at anything, this is essentially kind of like the conceptual framework of building an in silico brain model. Now let's link back to this. So can we simulate a fly connectome to predict neural activity? So if you recall the problem, so we know that the connectome shows a detailed map of how neurons are connected. So you see they reconstruct some kind of neurons, they take a bunch of slices and then they essentially create like neuPrint and Neuroglancer or whatever. So we have a detailed map of how neurons are connected. However, making equally detailed measurements of the dynamic or functional properties of each neuron and synapse in the circuit is not yet possible. Because we have 50,000 neurons, and in fact, experimentally, we only have uncovered like only 10% of this optic lobe so far, and that's why this data set is really impressive. We just don't know much about these neurons. So the point they're trying to make is, although we have a structural map, we do not fully understand the dynamic properties of each neuron and each synapse in a circuit. And it's also not maybe one day is possible, but in the near future, for sure, we will not be able to do this for all of the entire brain or even the entire optic lobe. So the question is, using this dead fly brain wiring diagram, how can we infer the computational dynamics of neurons and synapses to accurately predict neuroactivity? In other words, the computational dynamics of neurons and synapses is essentially that equation that I showed you. So that's the computational dynamics of neurons and synapses. Can we somehow do that to accurately predict neuroactivity? So when we talk about predicting neuroactivity, we're comparing to like well-known experiments. So now we're building a computational model and we see if this is kind of like similar to how the fly would actually think, I guess, in like real world scenarios. But let's see, how do we actually study this? Again, it builds on the framework that I've just talked before with slight tweaks. So the first idea is you build a neural network simulation of the fly's optic lobe, which again is the dataset that we're using. So we are building a neural network simulation of fly optic lobe with the same wiring diagram as that of the real fly brain. So in other words, you're creating a neural network model or some kind of computational model that respects the anatomical connections of the brain. So this is called a connectome-constrained network. I'm gonna repeat again. So we're gonna create a computational model that's basically based on the connections between the regions in the brain, right? So you take the fly brain, the optic lobe, you create a model using some kind of computational equations. That's essentially what this means. It's a connectome constrained model, a connectome constrained network. Just to show you what that means, so remember in our equation, so this is also the equation that Dr. Turaga used in his neural network paper. Essentially, we're using the information from, let's say, neuPrint or the fly optic lobe to inform this parameter. So remember, if you recall, this parameter is the known parameter. Why? Because we have this information from the structural connectome. Okay? So this is what it means to be connectome constrained. But remember also that we have a lot of things that we don't know. So if you recall the problem says that making equally detailed measurements of dynamic properties of each neuron is it's not yet possible. So essentially that is the free parameters. So this model essentially will have free parameters that correspond to the unknown dynamic properties of each neuron and synapses in each network. So essentially here, if you recall, these two, which is the time constant and the activity at rest or baseline, these are the unknown parameters. Essentially, these are the intrinsic properties of the neuron that the structural connectome cannot, we don't have information about that. Then, this is where it gets exciting. Then Dr. Turaga used deep learning, which is a kind of AI algorithm, to search for optimal parameters that allow these neurons in the simulated network to detect visual motion. So essentially, now you're building this whole brain model, and just like the real brain, now we can use this neural network model constrained by the connectome to detect motion. Okay, so that's very cool. So we're using deep learning to search for optimal parameters or the parameters that are unknown. We are searching for the optimal parameters that enable neurons in the simulated network to detect visual motion. Remember that these free parameters or these parameters, they are the simulated dynamic properties of each neuron and synapse in the network. In other words, these are not real. These are not experimental. These are the things that we have come up with. It's a simulated one. The big question is, do these simulated neuronal activity roughly equates to experimental neuronal activity? In other words, can we use deep learning to build simulate neuronal activity that can essentially predict experimental neuronal activity, or that's essentially the same as experimental neuronal activity? So how do they do this? They compare the simulated neuron activity patterns to the real experiment recordings from fly neurons across 26 experimental studies spanning 20 plus years. So the reason why they chose motion detection because it's very well studied in the fly. And so that's why they were able to compare this simulated network to the real experimental findings. And they found that the simulated neuron activities match really, really well with the real neuronal activity from these experiments. So essentially, they were able to create a connectome constrained networks that can predict neural activity across the fly visual system. But you can imagine that a lot of other behavior, like memory, for instance, is not very well characterized, like the fly visual system. So you could think of like, okay, now we can build some kind of connectome constrained network or connectome constrained model to now hypothesize how the neural activity would look like at different parts of the brain for different behavioral tasks. So this is really the cool thing about the connectome constrained networks. And essentially now you can do it for neurological disorder, right? You can create some kind of model for, let's say, epilepsy or Parkinson's disease. You can create this network to predict how this neural activity would look like in Parkinson's patient, and then that would inform the kind of treatment like deep brain stimulation where you kind of stimulate like a certain areas. So you can see like this kind of modeling of the brain using the connectome, let it be micro scale or macro scale. They're super helpful in understanding how we think, but also in helping treatments. So thank you, everyone. I wanted to ask if you guys have any questions. I know that was a dense talk, but I'm happy to stay on for 10 minutes.
Excuse me, but
Yes.
now have clap.
สวัสดีครับ
Uh,
Hello.
Hello. On behalf of Satik Tula, I would like to say thank you for your wonderful lecture. It was very knowledgeable for us to learn about connectomes and synapses and all sorts of things. So very appreciated.
Thank you. I'm glad you guys took the time out of your schedule to come and listen to something that I think is very fascinating, but also it's quite complex. But I hope you guys can join journey with me to learn about the brain and not just compete, but to just learn about neuroscience in general. I think that's the point of all of this. So if you go through the journey to explore the brain, you can essentially become a better neuroscientist, and one day you can explore this from the comfort of your home. So yeah, thank you.
I have a question regarding the part about the talk that you said. When you do modeling, you have to have like the anatomical constraint, and another one is mathematical description of the system. But when you said about the macro scale anatomical structure, like I have a question like how can we come up with such macro scale connection? Isn't it like I have heard like most of the macro scale connection mostly come from the functional connectomic, like maybe through like fMRI. But isn't it like if it like come from the functional mapping, how does it going to tell us
Yes.
about the anatomical mapping?
Excellent question. I love the question. So essentially, we do have a macro scale structural map, right? So essentially, that's the same thing. So actually, I wanted to show you another thing if I had the time, but I guess you asked the question. So in humans, there's a really recent field which is at first in human neuroscience especially, or actually even in circuit-based neuroscience for like a more micro scale stuff, we look at specific brain regions. So you remember like, okay, hippocampus in humans, which is responsible for memory. So humans, hippocampus in memory activates when we're trying to acquire some kind of memory, right? But then people sort of realize like, hey, the brain is way more complex than that. It is very important to look at the more like network level kind of thing. So more of like large scale perspective. So there's this researcher called Sporns Graph Theory Network Neuroscience. I think it should show up. It's one of these, yeah, it's this one. So this is one of the first papers. I mean, there have been works, but this is like a very cool review on like network science, like using graph theory and thinking the brain is like a complex structural and functional system. But why am I saying that? Essentially, if you look at this diagram, this is how we can do it for humans. So I'm gonna scale in here. So remember the first step of this framework is that you have an anatomical connectivity map. So this is what we know. Actually, we know everything. But let's for now assume that for the purpose of our model, we only take in the anatomy, the macro scale structural connectome. So you get the structural connectivity. And now this is the kind of like the matrix you see. You then use some sort of principles of physics. So in microscale, you use that neuronal kind of wiring thing, right? The neuronal firing model, I think that's what it's called. But here, actually, very recently, Gustavo Deco and Morton, who's actually my PhD supervisor, so they found a way to use statistical physics, so more like a macro scale perspective, to have different kind of a physics model. So the model that a lot of people use is the Hoff model, but essentially it just takes in some kind of like frequency data from the functional connectome, but it really doesn't matter. The point is that you using the structural connectome, you essentially try to simulate some kind of anatomically constrained functional data. Okay, so this middle part here is the simulated anatomical constrained functional data. Now, just like before, you now try to fine tune the unknown parameters. There are a lot of things we do not know about the structural connectivity, right? So we then try to fine tune these parameters in the Hoff model, the Ising model, or the DMF, which essentially what it does is it fits to the functional data. So oftentimes when I go to a scan, right? I would take a structural scan. I might even do a diffusion tensor imaging scan, but essentially you just need a structural scan of your brain. And then you take a functional MRI scan when you're at rest or when you're doing some task. So empirical means experimental. So just like you said, they have found a way to actually not require information about individual neurons, but instead they kind of try to use statistical physics and some kind of like supercritical system in the brain to kind of simulate the anatomically constrained functional connectivity, which then they fine-tune to fit to the real functional data that we collect on that individual human. And that's how we build a whole brain model for that specific patient or that specific participant. And because now we have the mathematical model that's anatomically constrained, you can do all sorts of cool things. Like you can perturb the system, right? Like you can be like, okay, what if I just add some random noise into this part of the brain area? Would the information about memory just disappear? Or you can answer like cooler questions like consciousness. Like a lot of time consciousness, it's a bit hard to study using regional analysis, but now you have like the whole network. You can study cool things like consciousness, like can perturbing some system actually leads to breakdown of this entire network and so on. I don't know if that was clear, but that's kind of what we do for the macro scale thing. Did that make sense?
Oh, yes, thank you. Oh, but for the parameter that we tuning in the macro scale, it would not directly reflect like the physical property of the neuron, right? It may be like a statistical mean of the whole region, something like that.
Yes, exactly. Yes, it would be a region to region analysis. So for instance, like this, it's very likely we break it down to, I don't know, 80 regions or like 90 regions, and then you kind of build the model. So you're right, yeah, you can't actually see neuron to neuron. But the beautiful thing about this is they found that because the brain operates at some kind of like macro scale system as well, so actually you could create anatomically constrained network without using the individual properties of the neurons. Because again, we have 60 billion neurons in the brain. So right now for humans, and also partly because we don't have information about individual neurons in the humans, so that's what we are doing at the moment. But hopefully 10 years down the line, we can like integrate the synapse level information to the human brain. Cool. Was that clear?
Yes, thank you.
Yes. And actually, I want to show you one more thing because I know a lot of high school students like to do research. So we have created a page for educators, but essentially you can have a look at it. Here, we try to map the combination to real learning objectives that are often done in like high schools. So, I mean, even if you are a student, you can look at this of how this can actually help you. If you are doing for an international program like IB Biology, it really well maps to some of the content in IB biology syllabus. But really, I think with any high school curriculum or undergrad curriculum in general, like the competition kind of works. Remember, you can also use your analysis to fulfill any kind of school-based research requirement. Oftentimes, access to experimental equipment might be a barrier. If you have a research requirement, having access to such a data set. So I think this is a very cool way to explore and do really cutting edge research in neuroscience from the comfort of your home. So feel free to use part of your submission for any of your school-based research requirements or for other competitions and so on. And so did we have a question in the chat? Yes, feel free to summarize this workshop for your friends. So someone asked, would it be all right if I later summarize the workshop for my friends at school in Thailand or share some parts of the workshops in Thai or in our school club? Yes, this is a good point. So I'm actively partnering with high school clubs in different parts of the world. For instance, we have partnered with a club in Libya, which is a country in North Africa, where they hosted a version of this workshop in Arabic and over 150 students attend. So if you're, I don't know if Satich have a high school club, but essentially you guys could partner with FlyComp 2025. We recently just sent a lot of outreach to high school clubs or societies. If you are interested, you can like email me directly. Yes, and this also might be a nice way to kind of work closely with our team and also inspire other students from your school or from other schools as well to learn the Connectome. Thank you so much, everyone. Have a good day. Bye-bye.
Clematis Research Empowerment Hub. Empowering research for everyone.